Alright guys, so today we'll be discussing about the germ layers. So what are germ layers? Whenever we talk about the germ layers, right, we have to go to the second week, and that's how we're going to talk about the first, right? So as you know, as we talked in previously, that you have this cell, this outer cell that I'm showing you this. This is basically called what? Outer, let's say, cell mass, right? And then this is called inner, let's just say, cell mass, because this inner cell mass, which is actually going to give something called the embryo proper, and later that's going to make embryoblast, right? And this outer cell mass, which is actually going to make the trophoblast, and later that's going to form basically trophoblast, and that's going to later form a placenta, right? That's something that you have to remember. Now, after this, let's erase this. What is the most important thing you have to remember here is that this inner cell mass, right? This inner cell mass, basically they get differentiated in second week, right? They basically form something called bilaminar disc, right? And this is a bilaminar disc. And the way they do it is that they express like different types of genes, right? Some of the genes will get activated. Like for example, there are some genes that will get turned on for the epiblast, and those genes will actually inhibit the other cells within this internal or inner mass. And what is going to happen is that can lead into formations of the another cells, these black cells, right? These cells that's in blue, right, which are columnar in nature, these are called, and this is happening in the second week, okay? And this is going to make something called epiblast, right? And then these cells right here, what do we call? We call them a hypoblast, all right? This is a cuboidal cell in nature. Now, after these formations of this, right, what is going to happen to this outer cell mass? They also go a process of, let's say, mitotic divisions, and ultimately what happens is that they'll make different layers of the cells like this, right? And then they will have this outer layer cells. What is going to form? They're going to form something called cytotrophoblast. Like this, we can call this as a cytotrophoblast. And then this outer layer, right, which are multinucleated cells, we can call them a syncytiotropoblast, all right? Syncytiotropoblast. Syncytiotropoblast is very important for actually giving a rise to B, C, beta, human choriogonadotropins. Now, these all inner cells, like your inner mass, there's the formations of, as we said, there's two cells right here, right? What is this called? Epiblast. And then the other one is basically a cuboidal cells, right? Now what I'm going to do is that I'm going to actually only make these two cells, okay? So basically I'm going to take this. I'm not going to talk about the other, this syncytiotropoblast, but I'm going to draw these cells right here. Okay, actually let me draw the cells right here. So if I make the cells, right, you basically have a cells looks like this. Let's just say that this is a epiblast, right? Epiblast cells, right? And then you have other cells right here, which is let's just say cuboidal in nature, right? What happens? These are look, this is what it's going to happen. Now, this is what is called bilaminar disc, right? And these cells, this is going to happen in second week of development, right? And one of the most important things you should remember is that this epiblast cells, right? What happens? These cells, started like sort of like this hypoblast started coming together and started fusing with this this epiblast, right? Because these are cuboidal. They started, fusing with these cells and this cuboidal cells sort of get converted into columnar, right? And when they're doing that, when they're fusing together, they actually started creating basically a thickening membrane, right? And let me show you how that will look like. So if I make this cells right here, right? If I make this, let's just say cells right here, right? And then if I make this cells right here, right? And then if I draw this like this, and you have something called, let's just say this is a hypoblast cells, right? And when they fuse together, right, they make a very important, like a thickening membrane structure like this, right? This thickening membrane structure, this is what is called basically a procaudal plate, right? Or sometimes we also call them the buccopharyngeal membrane, right? Because this is what is going to later, let's just say buccopharyngeal membrane, because later this is what's going to be a future map. And this procaudal plate, right? Basically, that help us to determine which one is a cranial and caudal axis, right? So basically, this would be your cranial, right, head regions, and this is going to be your caudal axis, right? Because this is what's going to be, this is what is going to be a future like head, right? Head and neck, and this caudal is, this is going to be future like a lower limbs, right? Lower part of the body. That's what it's going to be. Now, what is the most important thing you have to remember here is that, this epiblast cells right here, right? What is going to happen? These epiblast cells, from the posterior end, there is certain like genes that gets turned on, like a noggin proteins, right? Or there's something called a bone morphogenesis protein. And then from the posterior end, if I quickly make this, right? What really happens here is this. From the posterior end of the cells, right? What happens is that they started making this, and let's just say this is again, here, you have this procaudal plate, right? Like this. And what is going to happen here is that they started making like sort of like a structure like this, structure. This structure is very important structure. What is the structure is called? And they also form something called this. This structure which comes from the posterior end, this is basically called primitive, let's just say primitive strake. This is what it's going to form. And this is buccopharyngeal membrane, and this is called your primitive node, all right? All right, guys, so here I'm showing an image that I got it from the Langman. And basically, if you look at here, right here, that, this cells, the green, you see this, these two cells are basically derivatives of trophoblast, right? Trophoblast go through a process of mitosis and gives two different types of cells, syncytial trophoblast and then cytotrophoblast. But if you look at down here, this is happening in the second week where this cells is an epiblast. This is the one that's going to give rise to all those three germ layers, like ectoderms, endoderm, and mesoderms, right? That's what's going to happen. And as we discussed, what really happens is hypoblast cells, which sort of like fuses with the epiblastic cells, which are more in columnar in nature, right? And they basically make something called oropharyngeal membrane, or also called the buccopharyngeal membrane. And if you look at down here, look from the basically the posterior end right here, look, this from the posterior end, right? The cells sort of like started migrating towards the buccopharyngeal membrane, but it doesn't get all the way down here. And basically this region right here, this is what it's called your primitive. Basically, this is whole is a primitive strake, and there is a primitive node that also forms. I've discussed this in a nervous system video also, right? So this is a quick review. Now what is happening here is that this is a primitive strake, right? And this inner layer of the primitive strake, right? They also go degenerations, right? The process of degeneration so happens. And when that happens, what is that going to lead into? If you look at the structure right here, like you have this cells, you have this cells, you have this cells, you have this cells, right? So basically you have it, and you have this cells. And then you have this, let's just say in black, right? You have this. Now, and then you also have this green layer right here. And remember, there's still another layer, which is a black cells. What are those cells? These are my hypoblasts, right? Remember, this is the second week of the development. This middle piece, when they go degeneration, right? This is basically, now we call them, what do we call them? We basically call them, this part we call them a primitive, let's just say primitive groove, all right? And this is called primitive pit, right? That's what's happening. Now, this is very important. The formations of primitive strake is very important, right? Because whenever this primitive strake forms, right, and then really later what happens is that primitive strake later, it's going to get regressed. Because if this primitive strake does not regress, then, it can give you a condition called basically sacrococcygeal teratoma. Okay, it can give that condition called. And we'll talk more about that in pathology. But now, let's just say that, again, you have, what is the cell in the black? You have a hypoblast cells are there, right? Like this. Now, what is going to happen is that, there is basically the cells from like all the way from all these first cells, epiblast cells, right? They started migrating towards the primitive group, right? Primitive group. And as they're starting migrating to the primitive group, what happens is the cells from here, right here, right? From the cells from here, and all the way started going, and it started going like downwards, right? That's what's going to happen. And as this coming downward, I'm going to make a different structure, right? I'm going to make a different. As this coming downward, right? What happens is that they'll replace this layer of the cells, right? So this one, or earlier this was the hypoblast cells, right? As it's coming out, they started making a new layers of cells, and I'll make this another layers of cells, right? They started making another layer of cells like this. And this layers of cells, right? And we'll make this as here, all right? They're making this another layer of cells like that. And this another layer of cells they make, we call them a endoderm, all right? This is called endoderm. That's what they do. And not only this, they'll continue, like look, they'll continue making, the cells will continue actually, going across some morphogenesis, and from this primitive group, cells keep coming down and making a different layers, right? So they make this first layer, which is called endoderm, and after that, above this endoderm layer, above these layers, and then below this layer, right? This layer, which is an epiblast, this layer, there's another layers of cell that they form, right? Let's just make this as a green here. And these cells, right, they form, right? This layer we call them, because of the middle between this layers and this layer, we call them a mesoderm, all right? Mesoderm. And the rest of the cells, which remain on the up layer, right? We no longer call them, we no longer epiblast, we call them, what is this called? We call them ectoderm. So basically formations of this 3-germ layer, right, which is ectoderm, mesoderms, and endoderms, it's called gastrulations. Okay, what is this called? This is called gastrulations. And remember this, very, very important, that all of these ectoderms, mesoderms, endoderms, right, they all are derivatives from the epiblast cells. It's not from the hypoblast. It is actually from the epiblast cells, right? Because what happens, as these cells are coming down, they're making a different layers, and they also replaces the hypoblast cells, right? And then, and in the cells that are actually formed between the mesoderms, between this first layers and then the third layers, it's in the middle, so we call them a mesoderms, right? That is what's happening. And if you look at down here, you basically see that, look at this, this is a basic primitive node, right? And then basically there's a different gene that get expressed, and because of those, and the cells started migrating from down here, and all the way started coming down, right? As it's coming down from this epiblastic cells, right, start making a new layer. And when it started making new layers, remember, that replaces these hypoblastic cells, and they make what? Endoderm. Right? That's what they're going to make. And in between that, there's also another layers of cell they're going to make, and that's going to be your mesoderm. I'm going to show the pictures to you in a minute, okay? In a second. So if you look at another picture right here, look, this is basically, you see this a primitive streak right here, coming from the posterior end right here, and obviously the primitive node here. Not only that happens, now if I have to quickly make another layer cell. So look, here, what is happening here is that making another here. So basically, you have this cells right here, right? And then we made it another layer cells right here, look. This is, what is the cell called? You basically call this as a endoderms, right? And then you have a layer of the cells, which is a mesoderm cells. This is a mesoderms. Look. And then you also have the posterior end, you have this, right? This is a, look, you have a, and this is a caudal end right here, right? You have there's a buccopharyngeal membrane right down here. This is enough green for you. All right, now, not only this happens, right? Look, as the cells from the epiblast, they're coming and migrating and coming to the primitive group and making this different layer, make endoderm, ectoderm, endoderm, mesoderms. On top of that, there's also one process that happens where some of the cells actually come from this primitive node right here, right? And then they started making, the cells started actually migrating and going basically upward, anteriorly and upward, right? And make some sort of like a tube-like structures like this. That's what they're doing too. This tube-like structure, this tube-like structure is what is called notochord. All right, this is also happening here. So there's a lot of changes that is going to happen, right? Now, if I draw this and if I make in a more, let's just say, in a cranial way, right? Like how is this going to look like? Let's just make this as a cells. We'll make this as a, you have a different cells, okay? Okay, I'll make this as a different cells, okay? And then you have a, right? And then I'll make this as a, look. What is this? All right, and this is how it's going to look like. And you have a green end, you have this cells right here. All right? And then you have a red, you have this cells. Right? Look, this cells. This is your ectoderm right here. this is your mesoderms, right? This is your endoderm, right? And look, this structure right here, this is your notochord, all right? This is your notochord. Now, when there's a notochord is there, you know this is what notochord is going to do? This notochord is the one that's going to induce the formations of, like, you know, this, basically this ectoderm, right? Because this ectoderm, it's going to induce the formations of something called neural plate, all right? And I've discussed this in detail in nervous system video, right? So this notochord, you have to remember the functions of notochord, right? Notochord, let me write it down here for you. It is very important concept. The notochord, like let's just say you have a notochord, right? It's going to induce the formations of let's say neural plate, and then that's going to make your central nervous system, right? Induce the formations of neural plate, which will eventually form the central nervous system. Or you can simply say notochord basically, you know, are important for what? For neurulation, right? Second, this notochord later, it becomes a remnant for the nucleus of the pulposus of the spine. Okay, these are the two things that you have to remember. And the way it does is that, you know, the notochord induces certain growth factors. Like for example, you have a fibroblast growth factor like 8, like a bone morphogenesis, or let's say protein like noggin proteins, right? So this is very important, right? And when they form, right, they started forming of something called, let's just say, like this. And this is, you can simply say this is like a neural plate, right? That's what it's going to do. Now, if I have to make this, right? Now let's just say that. This notochord, now what happens is, later as this forms the neurofold, like let's say neural plate, this neural plate right here, you know, this is gonna go process of like a folding, right? Look, and now let me draw this structure here. When they go to folding, right? As this go to folding, really what happens is that, let's just say that, look, and you have this cells, right? When they go to folding, really what happens is that this is what it's gonna look like. It's gonna fold like this, okay? It's gonna fold like this neural plate, like this. And when it's folding, right? When it's folding, what happens is that some of the cells on edges right here, they broke down, they degenerate, right? And when the cells degenerate, let's just write down here, this cell degenerate, right, in the green right here, right? When they degenerate, right, what happens? These cells are no longer called, these degenerative cells, look, let's just say, these new degenerative cells, which are migratory in nature, we call them a neural crest cells, all right? Neural crest cells. Now, these neural crest cells, and obviously you have in green right here, right? You have what is the cell? You have the mesoderms layer, and then you also have this layer, which is an endoderm layer, right? And down below, look what happens is that, you know, there's this layer, this cells, this is, what is this in green right here, in blue, that is hanging, right? That is called right here, you will see, this is your notochord. So look, this is your ectoderm, right? This cells, mesoderms, endoderm, notochord, and this is a neural tube, right? Because it's actually neural plate which is folding, and later it's gonna form a neural tube. Now, how is that gonna look like? Look, now let's just right here. If I quickly make this, right? This picture is something that you have to remember, right? It looks something like this. All right, look, you have this structure right here, right? And then you have let me make it in green, this structure right here. And then, right? And then you have a big structure like this, right? And then you have right here. Right here, and then, look, what is this cell? If you look at it right here, right, this is how it's going to look like. Because when they fuse together, right, some of the cells degenerate, and then when the degenerate cells will be like this right here, okay, around this, okay, I'll make a different color, like right here. This cells appears at the edge, right, and these cells are basically a neural crest cells. So what happens when they fuse together? This comes out, right, like this, you know, And this will be it'll look like something like this. This is a neural tube, and the edges, like this one, is basically neural tube right here. This is a neural tube. This is your mesoderm. This is your endoderm. This is going to be your ectoderm. And then this layer is your neural crest cells. Okay, so this is how all the cells who gives rise to different parts of our body, right? Now, so basically what happens is that when the notochord forms, right, basically that notochord, this is a notochord, right, that basically induce the formations of what? Neural plate. And this neural plate, this started going something called neural folding, right? As this neural folding goes, like really what happens, look at this. This is what this is a neural group, right? This area. This neural folding is happening. And as this neural folding happens, look at when the cells are coming together. Look at this is surface ectoderm, right? Some of the cells like this right down here, it degenerates, right? These cells degenerates, and then when they degenerate, they form something called, look at this, the neural crest cells, right? These are neural crest cells. And those cells, like these cells, actually comes out and basically form a neural tube, all right? And this is your notochord, all right? So you have to remember this. Now after this, now we understood that how this, the neural crest cells were generated. We know how the ectoderms are generated. We know how the mesoderms are generated, and we know how the endoderms are generated, right? Now, one of the things that you have to remember here is this, that, all of these are coming from the epiblast cells, right? Now, what you have to remember here is that, these structures, that this mesoderms, right? This mesoderm within the cell, the green cells right here, right? They go internal morphogenesis, all right? So when they go internal morphogenesis, right, what happens is that, if I quickly make this, right, really look, one, you have this structures, right? So what really happens is this, there's a different layers of cells that will form. And what happens is this, inside the cells, they go morphogenesis, right? And when do that, this will appear at something like this. And what is this? We have to remember this. And you have look, it will form like this. Okay, it will appear like this. So when this happens, and there's a different cell, right? This is your endoderm cells, all right? And then right down here, you have you have this cells right here. And this is called let's just say, what is that called? Notochord. And you have this red right here, which is your neural tube. All right. Now, this green is basically your mesoderms, right? This is your ectoderm, mesoderm. This mesoderm go internal morphogenesis, right? And when you go to the internal morphogenesis, right? This right next to this neural tube, this we give a different name. This is called your paraxial mesoderms. Okay, this is what we do. And then this middle layer, right? Because the middle layer, we call them intermediate mesoderms, all right? And this portion right here, right? Which is gonna go and actually go to this layer right here, okay? Because it's gonna go to form basically this and cover the amniotic layer, right? We call them, this is a somatic, right? Let's just say this is all of them, because they're in lateral, we call them actually lateral plate mesoderms. But within the lateral plate mesoderms, there are divisions, okay? If it's going and making the layers on top here, right? We call them a somatic mesoderms. If it's going to and make like a viscera, okay? Then basically we call them a splanchnic, okay? Splanchnic mesoderms, because it's a very important concept, because somatic mesoderms, okay, and splanchnic mesoderms are very, because this somatic is basically saying there's a parietal layer, and splanchnic mesoderm is a visceral layer, because the splanchnic visceral layer, this is what's going to make your heart later, right? So this is a very important concept you have to remember. Now, this is your endoderm, right? And then obviously neural tube is there, and obviously you also have a neural crest cells out down there. But now we have to remember this, right? The three main germ layers are your ectoderm, endoderm, mesoderms. Now, if you look at down here, look at this, how this neural crest cells right here, right? This is another image where the neural crest cells is basically from here, it's going to degenerate, and this structure is going to be neural tube, right? And then obviously there's a neural core down here. And this is the image that you want to look. Remember, you want to remember. It's basically showing you all the different layers. You basically have this neural tube right here. You have this notochord, right? And look, this is going to be a mesoderm, this is going to be endoderm, and this is this up layer, this is you're going to be a surface ectoderm. And this is the image that you really want to remember, all right? Now, again, another image that I'm showing you where it basically shows like, how different layers are formed. And the most important thing is that just remember that, how, they can even ask you to, label this. Sometimes, they'll ask you, they'll show you an image and they'll ask you like, which structure is that, right? But most of the time they'll give you more in a clinical vineyard, right? But it's really important that you go over these pictures and know what this different structures looks like. But an ectoderm, remember this ectoderm itself went the morphogenesis, right? And it gives rise to, that's right down here, ectoderm, right? Let's just say your ectoderm, right? From the ectoderm, which is the first layer right here, right? Because it from ectoderm is your endoderm, right? Then after that, what happens? It made neural tube, right? From here, neural tube. Second, what did it make? It also made, okay, in the neural tube, they went then a neural plate it formed, and then what happened? The neural fold, and when the neural fold process, right, some of the cells degenerated, right? And they are called what? Neural crest cells. And then third, and whatever remaining cells, we call them basically surface ectoderm, okay? We call them a surface ectoderm. That's what the ectoderm did. And now if we go back and talk about, let's just say, mesoderm right here, right? What happens mesoderms? Mesoderm went to something called internal morphogenesis, right? That's what it went. This internal morphogenesis. And when it did, right? Remember how it made? It made a paroxial mesoderm. That's what it made, the first layers. Let's erase this part, right? It made something called, let's just say, a paroxial mesoderms, right? Or sometimes we call this somites, all right? We call them somites. Second, it also gives rise to something called intermediate mesoderms, right? And then it made a lateral, right? Lateral plate mesoderms, right? And then third, which is pretty easy for us, just the endoderm layer, all right? So we have to know, now, we have to know, the derivatives of the neural tube, neural crest, surface ectoderms, mesoderms, and also the endoderm. And this is very, very important that you should know because they will ask you on this exams, all right? Now, let's go and start from, let's just say, the neural tube. Now, for that, what are we going to do? So we're going to erase this part, right? I'm going to erase this. And we'll do this part. Here, what happens is in the neural tube, what is neural tubes are doing? And let me draw a line like this, all right? What are the neural tubes are doing? Neural tubes are basically making what? First is like from ectoderm, right? From ectoderm, right? Giving rise to neural tube, all right? Or sometimes we also call them a neuroectoderm, right? So let's just say you have a neural tube derivatives, right? Neural tubes are very important for what? Making a central nervous systems, right? So we can draw, let's just say, let's draw a central nervous systems, right? So maybe let's just draw a central nervous system like this, right? Now, and then you have what do you have? Look, you have this, right? Now this is central nervous system. Now, neural tube is going to form something called central nervous system, correct? But what are the central nervous system consist of? Central nervous system consists of what? Brain. Look, you have a brain. So this whole person is brain, all right? That is going to make that. And look, it is also going to make what? 2 is your spinal cord, right? So that, it is your derivatives of neural tubes, your brain and spinal cord. You have to remember this, right? On top of that, if you look at the brain, what do they consist of? Brain has what? Brains are lined by what? Like cells, right? Like tight junctions. And what are those, the cells, which maintain the blood-brain barrier, right? What are the cells are called? Remember in the brain, right? We're talking about here. These are astrocytes, right? Remember, look, think about it. We're not trying to memorize you. We're trying to see how the neural tubes are formed, okay? The astrocytes are there. On top of that, look, blood-brain barriers in the brain are done by astrocytes. right? Which is one of the glial cells, right? On top of that, you know, there in the brain, you have a what? There's a myelin, right? Those myelin sheath are what? Lined by what kind of cells? Oligodendrocytes, right? Let's just say oligodendrocytes. This is what you see. So you have a brain, spinal cord, astrocytes, oligodendrocytes, right? These are derivatives of neural tube. What else these neural tubes give rise to? You have to remember. We know the fact that it's going to give rise to astrocytes, oligodendrocytes. But what about some of the cells, like for example, microglial? You have to remember that microglial is a phagocytic cells. That's on the brain. That microglial cell is an exception. That is basically a mesodermal derivatives. Those are actually migrated to the central nervous systems, okay? So basically microglials is a derivatives, all right? On top of that, when the brains are forming, right? You know, the brains are forming like telencephalons and there's all those prosencephalons, right? Mesencephalon, mesencephalon. So consists of all the brainstem, right? That's coming neural tube. On top of this, if you look at here, right here, you know, in the brain, right? What else you're having here is this. Right down here, you have all layers of the cells like this, right? And then you make a two pouch like this, right? In the brain, you have this one. You can call it. Let's say this is called anterior pituitary, and this is called posterior pituitary, right? Remember, this posterior pituitary, it is what is called neurohypophysis, right? We call them neurohypophysis. This is also derivative of this neural tube. So this is posterior pituitary. Let's just write down here, posterior, or let's just say neurohypophysis, right? That is also derivative of neural tube, right? That's all. On top of that, look, what is happening on, look, there is a hypothalamus right here, right? There is eyes is also there, right? Look, this is what, you can simply say this is your frontal brain, this is parietal, this is, let's say, occipital temporal, right? There's eyes, right? And eyes are what? Give rise to your optic nerve, right? And retinas are also what? Derivatives of neural tube. So if you want to remember, neural tube derivatives, remember, think about central nervous systems, think about what, you know, the derivatives of this, optic nerve, retina, you talk about brain, you talk about astrocytes, you talk about the posterior pituitary, okay, neurohypophysis. So these are some of the most important structures of the neural tube, all right? Now after that, let's talk about the neural crest cells, all right? When you talk about neural crest cells, think about these are migratory in nature, okay? Think about these are peripherally located, or think about peripheral nervous systems, okay? Think about this, peripheral nervous system, right? And for this one, okay, these are found in all of our body, all right? Now, you have to remember, why do you usually your neural crest cells are found? That's something that you have to remember. And for that, you know, I got this mnemonics from first aid book. You can use it actually. You have Elmo, all right? And you have a passes. All right, this is a mnemonic for this, all right? But now let's just think. Let's try to draw where peripheral nervous systems are located. Peripheral nervous system basically means what? It's outside of brain, right? That means the peripheral nervous system consists of what? Think about it. Outside of brain, right? Peripheral nervous system. And whenever I think about peripheral nervous system, I think about, look, autonomic, right? Autonomic. Meaning you're talking about what are you talking about? What am I talking about? Look, you're talking about the ganglions, right? You're talking about the cranial nerves, right? So you're talking about, let's just say, you're talking about the cranial nerves, right? You're talking about the cranial nerves, right? You're talking about the autonomic nervous system, like sympathetic nervous system or parasympathetic nervous system, right? So you're talking about sympathetic, let's just say, and you're talking about parasympathetic nervous system. You're talking about the ganglion, but you're talking about dorsal because it's peripheral. Dorsal root ganglion, right? You're talking about that, right? And look, that's what this P is referring as, peripheral nervous systems, right? Now, what else is there? Look, what about this A? E is referring as. This E is basically saying it is derivatives of. Okay, let's just write down. That E is saying, look, let's just say this is one, peripheral nervous system, this is one. Second, these are basically enterochrophene cells, all right? What about the L? You know the brain, remember this brain has a layers, not the internal. There's different layers, right? You have different layers of this. You have, what are those 3 different layers of the brain? Meninges, right? You have a dura matter, you have organoid matters, and you have pia matter, right? Remember this. You have to remember this. This peripheral nervous system gives rise to the pia matters and arachnoid matter. What do we call them? Whenever there is a meningitis is there, whenever we're talking about meningitis, we always think about inflammations of the what? Inflammations of the pia and the arachnoid matter, not dura matter, okay? You have to be very, very clear about this. So think about this. Meningitis is sort of a misnomer, and it should be called leptomeningitis, because lepto basically referring as the arachnoid matters and pia matters. So these guys give rise to leptomeninges. Okay, let's just write it in black. And what are leptomeninges? They're nothing but leptomeninges, right? They're nothing but pia matters and then arachnoid matters. That's why this L is referring, right? What about this M is going to? Remember what M is basically telling you? M tells you something called what? M is referring as, it's gonna give rise to, melanocytes. It's very important. So that's M is going to be, okay, right down here. I'll write down in blue, melanocytes. All right? This is something that you have to remember. Now, what about this 5 right here? This is O, right here, O. O is basically odendoblast, okay? Odendoblast. Number 6 right here. This P, we talk about peripheral nervous system, right? Look, as we said, these neurocrystals are migratory in nature, right? Some of the neurocrystals, they actually migrate to these beautiful locations. And this is basically, if I have to make it, right? Look at this right here. And then this area right here, right? What does this look like? This is your adrenal, right? Adrenal gland. But the adrenal gland has 2 parts, adrenal cortex and adrenal medulla. This adrenal medulla is the derivatives of the neurocrest. Remember, it's adrenal medulla. And sometimes they ask you in these exams, you don't remember this, okay? Adrenal cortex, it is not coming from the neurocrest cells. Instead, that actually arises from some other area, right? And from where? From the mesoderms. Right? Something that you have to remember. So adrenal cortex is basically comes from the mesoderms, right? But adrenal medulla from the neurocrest cells. Then what about let's just say, what about this as is referring as, like a number 7 right here, okay? That is basic, look, you can simply say these is, cells that actually, these are cells which are, if I make this, let's just say, this cells, you have and then, it's going to go to, what is this? Central nervous system right here, right? The periphery ganglion right here, right? But these cells, right, from the peripheral nervous system, they're myelinated, right? These myelinated cells are, what do you call them? Swan cells, right? So those are called swan cells. So swan cells are what? Coming from, basically, neurocrest cells. Now let's just talk about 8. What is 8? If I make this, let's just say this is a heart, right? This is a heart. In the heart, right, there's a formations of a 2 septum, which is basically called aortico and pulmonary septum. Sometimes we also call them, let's say, spiral membrane, right? This spiral membrane, right? They are very important for actually, giving rise to the aorta and the pulmonary trunk, right? And neurocrest are very important actually for giving rise to that. So it is very important that you remember that, right? The aorticopulmonary septum. On top of that, there is also in the heart, there's also one more thing that play a role. Remember, the formations in the heart, there's also requiring of something called endocardial cushions, all right? Because if there's a problem with the endocardial cushions, the neurocrest problem, then it can give you something called ventral septal defects, all right? The ventricle septal defect, you can have that, right? So that endocardial cushion is there, important for that, right? So remember, look, what else we said? Look, let's repeat quickly. So you have neurocrest cells, right? Which are very, very important giving peripheral nervous systems, right? And those peripheral nerves in the periphery, what are they looking at? The cells, the nerves are myelinated by swan cells, the cranial nerves are there, the parasympathetic nervous system, dorsal root ganglia, leptomeninges, you have melanocytes, swan cells, you have aorticopulmonary septums, that is there, right? On top of that, you have endocardial cushion is there. These are basically all the derivatives of What? Neurocrystals. What else is there? Look, you have this S. What is this called? 10. It's called membrane too, right? It's called membrane. This also give rise by the neurocrust cells. Most important things that sometimes I've seen the questions they ask you, right, about this, especially this adrenal medulla, because there is a clinical condition that are associated with this, pheochromocytoma, right? So make sure that you remember the adrenal medulla is due to the neurocrust cells, right? And also the dorsal root ganglia and the leptomeninges. Now, after this, now let's talk about ectoderm derivatives which give rise to this guy, right? What is surface ectoderm, right? And for that one, let's erase this part right here, right? Let's erase this one. Surface ectoderm. And remember, surface ectoderm, we're referring as the, on the surface, meaning the outer covering we're talking about, outside, right? Surface ectoderm. Let's just say you have this important concept here. This is a surface ectoderm. Let's just write down here. You have a surface ectoderm, right? Or you can simply think of it as outer covering, right? Outer covering. It's located outside. Let's go with here. If you look at me, right, what do you see? You basically see what? Hair, right? So outer covering, you see hair, okay? This is what you see. Second, right? You can see skin, right? Skin, that's what you see, right? Skin, and then, skins, and usually uppermost layer of the skin, what do you call them? Epidermis, right? Epidermis. Because if this is skin, this outer layer, the most layer is called epidermis, right? Now, on top of the skin, the hair, look, what else you see? You can see an ear, right? External ear. That is also derived because it's surface outer, right? External ear, okay? Even internal ear. Now, if you look at it, look what I'm touching here. Nose, right? There is a derivative of nose, actually, this basically nasopharyngeal pouches, or let's just say from this surface epithelium of the nasopharynx, whatever, they actually migrate towards this guy. Remember here, we said this one right here. What did we say? We said this is a posterior pituitary, right? Posterior pituitary gland. What about this anterior pituitary gland? This anterior pituitary gland is actually comes from a derivatives of the surface epithelium of the nasopharynx, okay? So that give rise to anterior pituitary, right? And that is, what is that called? We call them obviously adeno, okay? Hypophysis, or we also call them anterior pituitary, or sometimes they can ask you and they can call you what? Rathke's pouch, all right? Sometimes they also call this a Rathke's pouch. All right, and this is something you have to remember because there's a clinical condition associated with the Rathke's pouch, okay? Adenohypophysis. Because if there's a tumor in there, right, that can give you a condition called bitemporal hemianopia, all right? So this is something you have to remember. On top of this, look, this is 5, 6. Look, what do you see here? Teeth, you see, right? The outermost covering of the teeth, what do you call them? You call them animal of teeth, right? This is what you see. So look, hair, skins, epidermis, external ear, adenohypophysis, animal of the teeth, right? These are all basically what? Derivatives of surface ectoderm. But if you look at the face, not only you see the animal teeth, right? You also see the gland, like you can actually feel it, like parotid gland. That is also coming from, let's just say, parotid, right? Parotid gland is also derivatives of surface ectoderm. So this is how you can actually, without memorizing this, right? Without memorizing this, you can actually distinguish which is surface ectoderms. All right, now, after this, now we're done with this ectoderm. Now let's go and move to this part, which is our mesoderm, right? And for the mesoderms, let's draw it down here. Let's erase this and we'll do mesoderms. And let's just say what the mesoderms do. Think of meso referring as what? Middle place. It's referring as what? Muscles. That's what it's going to give rise to. That's what we're talking about here. So if we talk about mesoderms, like let's just say here, right? There's a mesoderm, right? The derivatives of. You have to remember we said it's paraxial mesoderm, which are somites, right? These are somites. These somites can give rise to basically, look, you can remember this as this. You can remember as this. S, you can remember as, okay, let's just write. S, you can remember as M, and you can remember as a D. What is S is referring as? Sclerotome. What is this M is referring as? Myelotome, okay? And this is a dermotome. Look, what sclerotome means? This is supplying, okay? This is giving rise to, very, very importantly, you're talking about the ribs, okay? You're talking about the sternum, all right? You're talking about the ribs, sternums, you're talking about the vertebrae. This is what it's going to give rise to. If we talk about myelodome, it's giving rise to the muscles, right? Like for example, muscles of your, let's just say trunk, right? Trunk, body, right? And then limbs. And what are those muscles, right? Like for example, you're talking about the skeletal muscles, right? You're talking about the cardiac muscles. You're talking about the smooth muscles. It's giving rise to those. And this dermatome is referring as the dermis of the tissue. Remember, the epidermis is coming from the surface ectoderm, right? But then right below the epidermis, like let's just say, if you're talking about, let's say this is a epidermis, the below the cells right down here is, what is this called? This is called dermis, right? And that is given by what? Dermatome, right? This is so much. Now then, you also have the intermediate mesoderms, right? Intermediate mesoderms are very, very important for giving rise to what? Like if you want to write it down here, okay, let's just write down here, intermediate mesoderms, right? You have the intermediate mesoderms. And these are very, very important for making what? It's very, very important for making your kidney, right? Urogenital system, right? Kidneys. That's what it's going to give rise to. One of the exceptions you have to remember is that, you know, it's going to make kidney, it's going to urogen. They also make, look, I'll draw it here. You know, okay, what is this? This is your kidney, right? But what sits on top of them here is basically your adrenal gland, right? But this medulla is coming from neocrus, but this adrenal cortex, right? This adrenal cortex is actually derivatives of mesoderms. Everyone. On top of that, there's also sometimes you see a, you know, if let's just say this is a, let's say that this is my, what type of kidney? Okay, so I drew in a different color, but just remember, okay, let me draw the same color. All right, if it's a kidney is there, all right, let's make this as another here. Here, you have a adrenal medulla, right? But there's also one organ right here, which goes and maybe, let's just say, sits down, right down here, okay? What is this called? Spleen, right? And sometimes they ask you these questions, okay? The spleen is actually coming from mesoderms, right? This is something that you have to remember, because sometimes they ask you if a spleen is derived where? It's the mesoderms, right? Adrenal cortex, you have a kidney, right? Now after that, look, we talked about, what about, you know, we said this ribs and bones and sternums and body. That means what are we talking about here? Not only this, like you're talking about the basic musculoskeletal system, right? That's what you're talking about. It's give rise, the mesoderms are very important for giving rise to musculoskeletal system. Like for example, these guys, right, you know, you have a scrotum, vertebra, ribs, obviously the adrenal cortex, spleen is there, the urogenital systems. For the ribs and bones and vertebra, right, they have what? Cartilage, right? You have a cartilage is there, right? Cartilage, connective tissues. They're coming from mesoderms. Like for example, other one is like a phagocytic cells, like a microglial cells. This is important. Microglials are coming from mesoderms. That is coming from. And then after that, you also have now last is the lateral mesoderms, right? And remember, what is lateral mesoderms are giving? Lateral mesoderms are, remember lateral mesoderms, it actually gives rise to what? Somatic mesoderms, and then you have a splanchic mesoderms, right? So whenever you have a somatic and splanchic mesoderms, what are they going to give rise to? Think about this way. Lateral mesoderm important for, as we said, it's going to make a cardiovascular systems, right? And whenever you think of cardiovascular, what do you think about? Obviously it's cardiac muscle, but the blood, lymph vessels, right? Because heart is basically a pump, right? It's actually distributes the blood to the different systems. And blood, you have a lymph, okay, lymphatics. That is coming from the lateral mesoderms. And most importantly, you have to remember this too, that, you know, this lateral mesoderms, which give blood and lymph and all that. On top of that, look, on top of this, right, there is obviously lateral mesoderms, to be very specific, is the splanchic mesoderms, right? That is giving rise to your central nervous system, your cardiovascular system. We're talking about that, right? Basically, these are derived of mesoderms. Now, one more thing you have to remember here is that, you know, mesoderms also give rise to, remember how we talk about leptomeninges? it came from where? The neocrystals, right? But the outermost layer, which is a hard layer, right? Dura layer. That's why, dura matter is coming from this layer, which is a dura matter is actually coming from this point right here, is also coming from mesoderms. And to be very, very specific, when it comes to, let's just say, intermediate mesoderms, it gives rise to kidney. And, to be, rather than saying like urinary system, because let's not get confused, let's say gonads, right? Or ureter. Let's just say gonads. Let's just say gonads. It is coming from basically mesoderms, right? The gonads and then genital tracts are coming from the mesoderms, right? Now after this, again, quickly let's just say, if you want to remember something from mesoderm derivatives, you have to remember that, all the connective tissue like bones, muscle, cardiac muscle, smooth muscle cells, right? Those ones, like cartilage, those are coming from the mesoderms. And then it also gives us like your cardiovascular system, kidneys, spleen, you have to remember really, really well. Adrenal cortex are coming from the mesoderms. Now we're going to talk about the last one, and that is basically your endoderm layer, right? And remember, endoderms actually give rise to, lining of the epithelium, okay, of the internal organs, if you want to remember that way, all right? Let's just say this one, we've been doing this, this is, let's just say this layer is your endoderm layer, right? And this endoderm layers, right, this is giving rise to importantly organs right here. Look, what are the cells down here? Look, here the cells are, type 1, type 2 pneumocytes, who are actually, the type 1, type 2 pneumocytes in alveoli, right? They're actually lining the epithelium, they're lined with this epithelium. Those are coming from where? Endoderms, right? So this is endoderm, right? So basically lining of the epithelium, which actually gives rise to like a lower, like a respiratory tract, all right? Like for example, type 1, type 2 pneumocytes. Those are coming from this guy right here, which is an endoderm. Second, what is this? Right? And you have look, GI tract, right? Or we can simply say foregut, right? Lining of the foregut, right? GI tract. Or the midgut. And then hindgut, right? All of them are derivatives of what? Endoderms. And you have to remember, right? So GI tract, lining of that. So lower respiratory tract, like for example, your trachea, bronchi, lungs, all of them, right? And all of this, you also have to remember the genital urinary systems. Like for example, urinary bladder, right? Let's say urinary bladder. This is coming from the endoderms. What about if it's coming, urinary bladder is coming from endoderms, then what about the, let's just say the urethra or lower vagina? Those are also coming from the endoderm. So if you remember the endoderm derivatives, right, you have to remember the lower respiratory tract, that's a very important, alveoli, like bronchi, lungs, trachea, right? Those ones, lower respiratory tract, because they're lined with epithelia. You have to remember the GI tract, okay, GI organs. You have to talk about the midgut organs, and you talk about the hindgut organ also. And then on top of that, there's urinary bladder, urethra, okay, urinary bladder, urethra also supply or lined by the endoderm in air, endoderm. Now, the other things you should remember is that, there are organs like, for example, here there's a liver, the liver and parenchyma. There's also right down here pancreas, right? This liver, this pancreas is also coming from the derivatives of endoderm. Now, one of the other important concepts you remember is that there are the pouches, like pharyngeal pouches, right? And like, for example, the pharyngeal pouches like parathyroid gland, or let's just say palatine tonsils, right? The auditory tube, right? Those ones, right? Let's just write down the pharyngeal pouches. You have a pharyngeal pouches, right? Palatine like tonsils, right? Parathyroid, like basically you have a parathyroid glands, or thymus, importantly thymus, right? These are derivatives of endoderms. Endoderms, remember that I said the surface ectoderms are coming from the parathyroid gland, right? But there are other glands, like you see in here, like submandibular gland, or let's just say sublingual glands, right? Submandibular and sublingual glands that are coming from endoderm. So basically, there's a tumors of the, let's just say, submandibular gland. Let's just say you have submandibular gland, or like sublingual gland, right? These are basically what? They're coming from the endoderm in nature, all right? So, If you want to remember the endoderm, so you just think about they're lying the internal organs, right, of your GI tract, so like foregut, midgut, hindgut. Remember the lower respiratory tract, right, the alveoli, bronchi, they're lined by that. And remember this, like basically thymus is very important. That is endodermin derivatives, right? And those are pharyngeal pouches, right? The palatine tonsils are there. And on top of that, I really want you to remember the parenchyma that makes by the endoderms are submandibular and sublingual. This is a sublingual gland, but not the parotid gland, because parotid gland is actually it is derived from surface ectoderm. All right. Now, one of the things that let me quickly tell you is that, you know, like you have all the serous membrane, like the parietal layer and visceral layers on your body, like serous membrane. Let's just write it. Those serous, like all serous, are the body cavities. Remember, you know, your parietal pleura, right? The parietal and visceral pleura, right? Those are serous membrane, right? Those in your body, right? Those are derivatives of this mesoderm. And this is also something that you should remember. Now, this image that I got it from basically the first aid book, and here it actually basically tells you the derivatives of, let's just this ectoderm, which is the surface ectoderms, neurotubes and neurocrystals, right? All of them. And then there's also the structure that actually is given by mesoderms and endoderms. There are certain, like for example, cells that probably I didn't talk about it. Like for instance, for endoderm, like the formations of gallbladder or pancreas, right? These are actually coming from endoderm. This is something that you have to remember. And very important, like, you know, always remember that even though it's an internal organ, the endoderm gives rise to. In the endoderm, right, one of the most important you should remember, sometimes they ask is this parathyroid follicles and the parafollicular cells, right? These parafollicular cells are coming from the endoderm. This is very important that you should remember. And when it comes to mesoderms, right, you should remember the spleen, adrenal cortex, right? And obviously the microglia is very important also. And the neurocrystals, you should remember all the different types of cells, because all of them are extremely important, especially the atypicopulmonary septums, endocardial cushions, right? Because there is a clinical pathology related with this. All right. Now, remember, we talked about a lot of what it can give rise to, because, you know, it is important that you try to remember some of them, because they'll ask you in your step 1, their NBME exams, right? They'll ask you at least 1 or 2 questions on this. So make sure that you remember this, all right? And I try to draw it, I try to go over and try to repeat, so that way you can actually remember this information. All right, guys, so for the germ layers, I think this is all I have for you guys, and thank you for watching.